Hello, and welcome to this discussion on infiltrative gliomas. And this particular discussion will be based on the 2021 WHO classification, the fifth edition. And this is mostly supposed to be a postgraduate refresher. So we will probably not be touching on all of the pathologies, but mostly concentrating on the ones which we get to see clinically. So what shall we be talking about in this particular discussion? We shall be talking about the changed nomenclatures. As we know, quite a lot has changed over from the 2007 classification. So we shall be talking about that. We shall be talking about the various kinds of genetic and immunohistochemical parameters that we routinely utilize in order to address this particular diagnostic entities. So we shall be talking about IDH, ATRX, and also in the process, we'll be talking about a few of the genetic events, like for example, the TERT promoter mutations, CDKN2A and 2B homozygous deletions, etc. And in this process, we shall also be touching upon the histone gene mutations. So over there, we shall be talking about the two important categories of tumor, that is H3, K27 mutated gliomas, that is the diffuse midline glioma, and the H3, G34, R and V diffuse hemispheric gliomas. So as you can see, the WHO CNS Tumour Classification has had a long history of evolution, starting from the year 1979 and going on to 2007 classification, which is the famous fourth edition, which was revised in the year 2016. And now we are in the age of the fifth classification, that is the one that came out in 2021. So quite a few has changed over from the year 2007, and we shall be summarizing those changes in the next subsequent slides. The 2007 classification. Now, this is a particular classification that I have a fondness for, simply because this was the one that I read during my PG days. But of course, as far as the WHO history of evolution of the WHO classification goes, this one is pretty prehistoric. But I shall start off this discussion with some of the key points which were utilized for getting to the particular diagnosis that we gave out in those days. So I shall be mostly stressing on morphology in this next few slides before we hit on the road with IHC and the molecular markers that currently define these entities. So if you had a diffuse glioma, and if you had features like this, as you can see, these fibrillary astrocytes, they have got nucleomegaly along with nuclear coarsening. There's nuclear contour irregularity, and these are all the findings which we call astrocytic atypia. So the atypia in the context of the neoplastic fibrillary astrocytes would actually define a tumor as a diffuse astrocytoma, and we would call it grade 2. Add mitotic figures to this finding of atypia. Now, there was no absolute cutoff for these mitotic figures. In a small biopsy, even a single mitotic figure would do. In a small biopsy, even a single mitotic figure would do, while on a larger specimen, you would need more. Add this issue of mitosis, and from the diffuse astrocytoma category, you have jumped over to the anaplastic astrocytoma category, and that got a grade of 3. Add on some other important findings, and you will have one more jump onto the 4th grade. Now, this particular thing that you see, this is something that we call pseudo-palisading necrosis, or you don't always need pseudo-palisading necrosis. Even your diffuse geographic necrosis would do, but you will need to make sure that you exclude the cases which have received radiotherapy, because radiotherapy induced necrosis does not have a prognostic significance. So you have necrosis over here, and in the lower picture, you see proliferation of the vascular cells, but as you can see, there's a bit of the capillary lumens which are seen here, but there is a proliferation of the cells which surround the capillary lumen. And this particular type of proliferation is what we call the microvascular proliferation. And some people like to call it glomerular endothelial hyperplasia, but microvascular proliferation is a better description. So any combination of necrosis with microvascular proliferation or just microvascular proliferation alone would suffice to upgrade this diagnosis of anaplastic astrocytoma onto a glioblastoma, and that got a grade of 4. Coming to the oligodendrogliomas. So this is the characteristic appearance of the oligodendrogliomas. I shall be talking a bit more about the histological morphology in the subsequent slides, but you have this characteristic fried egg appearing round nuclei with the perinuclear halo, and you have the chicken wire capillaries and the calcification. There's not much of cellularity, atypia, or mitotic activity to warrant a higher grade. So this gets a diagnosis of oligodendroglioma, WHO grade 2. Add findings like hypercellularity, but most importantly, mitotic access, along with the glomeruloid endothelial proliferation, that is the microvascular proliferation, and you have a jump from grade 2 oligodendroglioma onto anaplastic oligodendroglioma, which gets a WHO grade 3. Now, this is an interesting tumor, and obviously, this particular diagnosis is absolutely wrong. You don't give, you don't give out this kind of diagnosis these days. Not in the days of ATRX and IDH and 1p19q co-deletion fish. But back then, when we did not have those things, there used to be those tumors where some people would say, okay, some of these cells do look oligodendroglial. They're a bit round with the perinuclear cytoplasmic clearing, and some people would argue saying, no, I think this is a astrocytic tumor. You see, the cells are too elongated, too pleomorphic to fit into your description of a atypical oligodendrocyte. So I think it's astrocytic, and at the end, nobody would reach a consensus. And the diagnosis that might be given would be oligoastrocytoma, WHO grade 2 to 3, depending upon whether you find mitosis or not. Now, this was the favored approach in the 2007 WHO classification. In the 2016 WHO revised classification, they had warned against this particular diagnosis, saying that if you have done your IDH and ATRX and 1p19q codal workup properly, then you probably should not be giving this kind of a nomenclature. And now we know that this is an absolute no-no. You don't label out a tumor as oligoastrocytic. Is either oligodendroglial or astrocytic, but you will have to have a robust workup in order to reach that conclusion. Now, from 2007, we have come a long way. We are in the space age of molecular pathology, and the current WHO classification, the one that came out in 2021, there is a 5th edition, stresses that. So in this current era, those obsolete terminologies, the ones that I spent some time in describing, have gone out the window, and you will need to ask a few questions. Like, for example, before calling something astrocytoma of grade 2, you will need to ask about what's the IDH and ATRX status, and whether a CDNK2A and 2B homozygous deletion has been assayed or not. Similarly, prior to calling something an oligodendroglioma, you would again ask about the IDH and the ATRX status, and also the 1p19q co-deletion FISH or NGS assay, whether it has been performed or not. Similarly, for a case of glioblastoma, you will need to ask questions about the IDH-ATRX status, also need to know about the histone gene mutation status. And in addition to that, you will also need to ask about a few of the other molecular genetic abnormalities, which will help in establishing a diagnosis, establishing a molecular diagnosis of a glioblastoma in a tumor which might not look high grade. These would be TERT promoter mutations, EGFR gene amplifications, and gain in chromosome 7 and loss on chromosome 10 copy numbers. So we see that there has been a shift. Back in those days, when I did my training, the focus was mostly on microscopic, along with a bit of IHC evaluation for things like GFAP, OLIG2, and Ki67. But now the focus has changed over from microscopy onto these two advanced methodologies. That is, one is the DNA methylation profiling, and most importantly, the next generation sequencing, or NGS. So the diagnosis which is given out nowadays is not entirely histological. It's actually a combined approach of histology with IHC and molecular genetics. So it's a histomolecular diagnosis that we are aiming for with the passage of time. The two important molecular genetic methodologies that we shall be talking about now, in this discussion, we won't be having time to talk in detail about these particular methodologies, but the basic approach is that you basically collect the DNA and then subject it to restriction enzyme digestion. Now I'm talking about the DNA methylation assay. Go in for a procedure known as bisulfite conversion, and then follow it up with a next generation sequencing or a DNA microarray. So, and this combination of tests yields a huge amount of information. It yields information about mutations, as well as about things which we call copy number variations. And the CNVs, or copy number variations, are very, very important in the diagnostic workup of brain tumors, as we shall see subsequently. So over here, you have a methylation profiling scanner, and this instrument below is the next generation sequencing machine, one of the machines in the portfolio of Illumina. The data that is generated after DNA methylation is uploaded to a website known as Molecular Neuropathology org. It's a website which is maintained by the University of Heidelberg, and they have got a huge database of around 28,000 tumor samples, and they have basically divided those tumor samples into cohorts, and the information that is uploaded by you onto the server will be compared against those reference cases at the German Cancer Research Center. And as a result, and finally, you will have a plot in which your tumor cases, like in this particular case, it was a study of 53 cases, and these cases will be basically plotted against the known reference cases at DKFZ, and will be plotted by artificial intelligence and machine learning. And as a result, the final result will be in the form of a methylation plot, wherein your tumor gets a particular diagnosis based upon which particular tumor this case fits into. So it's a kind of a clustering which is done by the machine, and unsupervised clustering, which will be later supervised and finalized out in the form of a report which will reach you from DKFZ. As you can see over here, the one in the pink is the diagnosis that was made in your lab. So the diagnosis that you gave was glioblastoma WHO grade 4. The methylation profiling was done. It was analyzed against the cohort, against the cohort cluster at DKFZ, and your the final report that you got was a methylation class family. It confirmed your diagnosis of glioblastoma, and in addition, it also gave you the finding that it's IDH-wildtype. So thus helping to further pinpoint the diagnosis of glioblastoma into a certain category of glioblastoma, which is advocated by WHO. Now, this is a very, very powerful tool, and most of the power of this particular test lies in the fact that it can assay for copy number variations. As you can see, if you see the copy number variation profile on the right, so you see that the ones which are towards the middle, the green and the red shades toward the middle, are basically the stable copy numbers, whereas a few chromosomes seem to have the copy numbers which have gone up, that is a positive gain, the ones in green, and those which are below the baseline, that is the ones in red. So these would be the amplified ones, the ones in green, and those would be a chromosomal loss, the ones in red, the ones that have dipped below the baseline. We shall be seeing a few more of those plots. Most importantly, these copy number variation profiles would help to change your morphological diagnosis. Maybe you have got something like a grade 2 IDH mutated glioma, and then you come across a finding like there's a deletion in CDKN2A, 2B. This automatically upgrades your diagnosis to a IDH mutated astrocytoma grade 4. Thus, this is a very powerful tool, this DNA methylation profiling. However, you need to keep in mind that it has not been clinically validated, and it's only used for research purposes, and these things have not come into routine clinical practice yet. But WHO seems to be stressing on that, and maybe by the next WHO classification, we shall see a routine incorporation of the DNA methylation profiling in the diagnostic workup of the brain tumors. So like I said, the copy number variation, or the CNV assessment, which can be done by DNA methylation profiling, is very important. As you can see, the first case is an astrocytoma IDH mutant grade 2, in which you have got a relatively flat copy number without much of gains, or that is without much of amplifications, or without chromosomal loss. So this fits in with the IDH mutant grade 2. That is, you don't have significant genetic, you don't have copy number variations which might actually upgrade your diagnosis to a higher molecular grade. But the case below, you're seeing that there are quite a few of the chromosomes which are showing amplifications. So there are amplifications of certain chromosomes. There are also deletions of a couple of chromosomes. But most importantly, at the CDKN2A and CDKN2B locus, there is no chromosomal loss. And that's important, because had that thing been there, then your diagnosis of astrocytoma IDH mutant grade 3 would have actually become a grade 4, like in the next case. For example, over here, we see that there are basically there are chromosomal losses and gains, but most importantly, there's a homozygous deletion of the CDKN2A, 2B locus. And this particular thing warrants a poor prognosis. So even morphologically, if this particular thing has a grade 2 or a grade 3 appearance, if this particular case has that appearance histologically, and your DNA methylation profiling turns out a report of the presence of homozygous deletion in CDKN2A, 2B locus, your diagnosis becomes astrocytoma IDH mutant grade 4. Note that this is just one of the ways by which you can reach the diagnosis of, you can reach the conclusion that there is a homozygous deletion of CDKN2A, 2B. In many labs, this issue will still be settled by FISH. What about diagnostic workup? I mean, what about reaching diagnosis of tumors like oligodendroglioma? We know one important defining feature of oligodendroglioma is 1p, 19q co-deletion, right? So over here, you see that in the chromosome number 1, there is a loss in chromosome number 1. So basically there is a dip below the baseline, below the stable copy number, whereas there is also chromosomal loss at 19q. So this means there is a homozygous, so there is a co-deletion of 1p and 19q, thus establishing a diagnosis of oligodendroglioma, IDH mutated 1p, 19q. This is grade 2. So this DNA methylation profiling has emerged as a very, very powerful diagnostic tool. And over the last 4 or 5 years, lots of new tumoral entities in CNS tumor lineage have got discovered simply by utilizing this powerful methodology. Like, for example, your diagnostic entities of MGNT, myxoid glioneuronal tumor, or infantile hemispheric glioma. Then your thing of CNS neuroblastoma, FOXR2 activated, or BCOR alteration associated neuroepithelial tumors. These are all diagnoses which have been settled by utilizing this powerful methodology of DNA methylation profiling. So as per the new WHO classification, you are supposed to give out a layered report, right? So the first thing that is to be mentioned is the site of the tumor. Like, for example, here is the pons. Then the third one is your histological classification, wherein you just give the diagnosis which you have arrived by a combination of histology with IHC, and the molecular information needs to be submitted at the bottom, wherein you will mention about the molecular methodologies which were utilized for you to reach that particular diagnostic nomenclature. In this particular case, both IHC as well as next generation sequencing has shown that there is a mutation in the histone 3 gene at the K27 position. So the final integrated diagnosis in this case becomes diffuse midline glioma H3 K27 altered. So this is the layered reporting structure. Now, obviously, this particular kind of approach helps in a place where you have got access to things like NGS and a wide array of IHC markers. In many cases, you will probably be stuck with the diagnosis of glioblastoma, NOS, because you don't have the resources to basically work up the case for IDH, ATRX, and the histone gene mutations. In this particular context, WHO has these two nomenclatures. It's got NOS and NEC. So NOS basically means that you don't have the resources in order to reach a WHO approved diagnosis, or maybe you have the resources and they don't perform well. And NEC, or net elsewhere classified, or not elsewhere classified, would be a category wherein you have those diagnostic methodologies, but utilizing them, you come at a result which is not there in the publications, something weird. So in that case, it would have the NEC appended next to its name. Now, as you see, the DNA methylation profiling, a very powerful tool. Basically, in this particular journal article, it has shown that lots of different tumors which were priorly diagnosed by a combination of histology with IHC later got upgraded or downgraded to another different tumor class altogether after utilizing DNA methylation profiling. So, for example, your something which had been labeled as glioblastoma got reclassified, that is downgraded to anaplastic pleomorphic xanthoastrocytoma, or maybe something which was given a lower grade diagnosis like a PXA or an anaplastic PSA got upgraded to a glioblastoma IDH-wildtype. And hence, this is a very, very powerful tool which has got the potential to basically upturn the way that we look at diagnostic neuropathology. And artificial intelligence and machine learning has got a very important role to play in this. Now, getting back to our original discussion, where have we come? from 2007 to 2010. So from 2007 to 2021, we have come a very long way. The nomenclatures that I had initially used of diffuse anaplastic glioblastoma of grade 4, there's the Roman numerals, everything has gone out of the window. We see a few salient changes which have happened. Firstly, the new terminology, the new WHO classification does not allow you to utilize terminologies like anaplastic, most importantly. Second, these Roman numerals are all gone. They have been basically changed over to the Arabic numerals. And that's important because sometimes while typing out a report, your something of a grade 2 might get a entry as grade 1. Okay, so that was one of the reasons why they changed over that thing. And most importantly, we see that this particular diagnostic entity that I was talking about, oligoastrocytoma, has gone out of the window. We don't have this particular diagnosis now. Instead of this, the diagnostic methodologies which will be utilized to reach the nomenclatures as advocated by the new WHO classification are IHC, wherein your lab will have to have a pretty strong panel in which you will need to have an IDH1 R132 ATRX, possibly P53, because it really helps. H3K27 and histone, H3K27 trimethylated histone, H3G34V and H3G34R. I shall be talking about all these things once I get to the point of histone gene mutation associated gliomas. And fish, obviously you will need a fish for detecting things like 1p19q co-deletion or CDKN2A2B homozygous deletions. But of late, this fish-based technologies are giving way to next generation sequencing and promoter methylation assays. So Sanger sequencing, which was utilized in the past, along with the next generation sequencing, will be needed to assess for IDH1, 2, and histone mutations, as well as TERT promoter mutations. And the copy number variations that I talked about, 1p19q co-deletion, homozygous deletion of CDKN2A2B, EGFR amplification, and gain in chromosome 7 and loss in chromosome 10, all can be addressed by next generation sequencing or by promoter methylation assays. Utilizing this approach, we see that the new diagnostic terminologies that we come across are astrocytoma of the IDH mutated category, which in turn includes grade 2, grade 3, and grade 4. Note that those tumors, that is astrocytoma IDH mutated grade 4 ones, might morphologically look just like a glioblastoma, but they will not be called glioblastomas. Instead of that, the term glioblastoma is meant for those tumors which belong to the IDH-wildtype category, and they get a grade of 4. Oligodendroglioma, the name stays the same, but in the nomenclature, you will have to add these terminologies that you have to have an IDH mutation detection and also 1p19q co-deletion at the same time, and it gets grades of 2 to 3. Coming to the histone mutated gliomas, you have got two categories, one in the midline location, which is the H3K27 altered category, and one in the cerebral hemispheres, which is the H3G34 mutant category. Both get grades of 4, very, very high grade, irrespective of however low grade they might look histologically. You have got diffuse pediatric type high grade gliomas, and in which the tumor is wild type, that is, it does not show mutation in either the histone H3 gene as well as the IDH genes, and it gets a grade of 4. And you have got this category of infant type hemispheric glioma, which is basically associated with your receptor tyrosine kinase abnormalities, and this is the last category of the diffuse high grade gliomas. So you see that we have basically come a long way, starting from those, those primitive terminologies that we used in 2007 to more specific tumor nomenclatures that are based upon utilization of things like immunohistochemistry, along with lots of advanced molecular genetic methodologies. Now, getting back to the morphological part of the discussion, let's look at this case. Slight amount of hypercellularity over here, moderate amount of cellularity, and you have got this pleomorphic fibrillary astrocytes, and this population of polygonal cells with eccentrically pushed out nucleus and atypical nuclear morphology and polygonal cytoplasm are the gemistocytic astrocytes. You need not really add those terminologies of gemistocytic or fibrillary to your final diagnosis, because those are just morphological findings, but it's important to note these things anyway while describing your tumor. So these cells are obviously pleomorphic, hyperchromatic with irregular nuclear contours. And back in the olden days, they would have been given a diagnosis of diffuse astrocytoma, grade 2. We know that now we don't, that is grade Roman 2. We will not use this particular nomenclature now, and the present nomenclature will depend upon the IDH ATRX results along with the gene sequencing findings. This case over here, you have the similar findings, but most importantly, you have an atypical mitotic figure. So that upgrades your diagnosis from a grade 2 to grade 3. Priorly, it was called anaplastic astrocytoma. We know that in this present day, we will not use the terminology anaplastic. Rather, we'll have to see whether the IDH and ATRX, what are the patterns of IDH and ATRX expression in this particular case, and also the gene sequencing results. In this particular case, we have a pretty hypercellular tumor, which is composed of pleomorphic atypical fibrillary astrocytes with prominent mitotic activity. Now, these are all findings that we saw in our prior grade 3 category of astrocytoma. However, added to that is this finding of pseudopalisatic necrosis, as you see. Okay, so you have got these prominent areas of pseudopalisatic necrosis where inside you have these necrotic these necrotic atypical fibrillary astrocytes, which are palisaded by a rim of preserved atypical astrocytes. So in the olden days, this would have been called glioblastoma WHO grade 4. But again, now you will have to work up this case for the IDH mutational status as well as the ATRX preservation status before you reach any diagnosis. In this particular case, you have the pseudopalisatic necrosis as we have seen priorly, but you also have these glomeruloid endothelial proliferations over here. So you see the structure looks like a glomerulus. This is basically what we call microvascular proliferation. So you have these small capillary lumens which are rimmed by a layer of endothelial, proliferating endothelial cells, giving rise to an appearance like a glomerulus. So this is obviously a higher grade finding, which takes your diagnosis towards a grade 4. But again, IDH and ATRX status detection will be important before reaching a nomenclature. In this particular case, you have got a high-grade tumor which is basically composed of atypical cells which are arranged in intersecting fascicles. And this is obviously a gliosarcoma of WHO grade 4. In this case, this also belongs to the glioblastoma category. This is a small cell glioblastoma, wherein these tumor cells are very, very small. They don't exhibit the kind of monstrocellular appearance that you see sometimes in case of glioblastoma. Rather, they could be sometimes confused with a oligodendroglioma of grade 3. But as you see, the mitotic activity is pretty high. And even a grade 3 oligodendroglioma will often not show that much of mitotic activity. This is a giant cell glioblastoma, as you can see, which is defined by the presence of this pleomorphic, almost anaplastic, actually anaplastic looking bizarre astrocytes. It can be sometimes confused with an anaplastic pleomorphic xanthoastrocytoma. Okay, so you have got these bizarre astrocytes of giant cell morphology. This particular variant is known as a granular cell glioblastoma / astrocytoma. So you have got a central area where you have got necrosis, which is basically rimmed by diffuse sheets of polygonal histiocytoid cells with abundant pale cytoplasm, giving it an appearance of macrophage. A problematic issue about this particular tumor is that actually CD68, which is a macrophage marker, can be positive. However, more specific macrophage markers like CD163 will be negative in these cases. And if you do a GFAP, you will see a positivity for GFAP that can be seen within these tumor cells, thus establishing a diagnosis of a granular cell glioblastoma. Now we shall be talking a little bit about the various gliomogenic events, because an understanding of those things is important when we talk about things like, ATRX and IDH mutation status and all that. So the entire glioma family comprises of mutations in various kind of combinations. So at the heart of it is IDH 1 or 2 mutations, which are present in most of these high-grade gliomas, except for the IDH wild-type glioblastoma category. ATRX inactivation is something which is present in the case of IDH mutated astrocytomas, and also in the case of certain histone gene mutation associated gliomas. 1p19q co-deletion, as we have said, is seen in the case of oligodendrogliomas. And aside from them, you have got the mutations in the promoter sequence of the telomerase reverse transcriptase and homozygous deletions of the tumor suppressor genes CDK and 2A and 2B. So this particular slide is busy, but just focus on the main things. This is about the role of the IDH mutations in gliomogenesis. So the role of IDH is basically that it combines with isocitrate and gives rise to alpha ketoglutarate, which is utilized for synthesis of acetyl-CoA, which in turn is utilized for, for the synthesis of lipids. But most importantly, it also generates NADPH, which protects the cell from free radical mediated injury. Now, in the IDH mutations, there are basically multiple types of IDH, but out of these, IDH1 and IDH2 are the most important. The commonest mutation is the IDH1 mutations, which is called R132H, wherein R stands for basically arginine. So arginine is replaced with histidine. But this is the commonest mutation that is seen in the case of IDH, and it involves the IDH1 gene. And the resultant protein, that is R132H protein, can be detected by IHC. So this is the commonest genetic mutation that you see. But sometimes you can come across less common mutations within the IDH1 gene, like R132S, R132C, etc. And these are known as non-canonical genetic mutations. And even less often, you can come across mutations in the other gene, that is IDH2. Okay. So although 90% of the cases of your IDH mutated astrocytomas will have IDH1 mutations, that the remaining will have your non-canonical mutations and IDH2 mutations. The important thing that happens is that with this mutated IDH protein, a portion of the NADPH and alpha ketoglutarate, which is synthesized priorly, gets shifted over to the generation of an oncometabolite known as D2-hydroxyglutarate. This in turn inhibits a demethylase enzyme, and this results in increased hypermethylation. And we know that hypermethylation is associated with a gene inactivation, right? So this hypermethylation causes inactivation of multiple tumor suppressor genes like P16, P14, MGMT, etc. And all of this gives rise to the first gliomogenic event. And also importantly, this oncometabolite results in increased DNA damage. I mean, it results in loss of the cell to recover from this DNA damage. So this is the event number 1, and this event is seen in the case of IDH mutated astrocytomas and oligodendrogliomas. So both of these tumor categories share IDH mutations. These most often are in the IDH1 in the form of R132H mutations, but sometimes they can be non-canonical mutations within the IDH1 or IDH2 gene. The second event is basically ATRX inactivation, ATRX gene inactivation with loss of ATRX expression. And this occurs only in the case of astrocytoma, IDH mutant, which ranges from grades 2 to 4. That means your oligodendrogliomas will show IDH mutation, but they will not show ATRX loss, while your astrocytomas of the IDH mutated categories will show both IDH mutation as well as loss of ATRX expression. We shall be seeing a few of these cases to bring home the point. So in this particular case, the initial diagnosis was a grade 2 astrocytoma, which recurred over a period of years, and then the genetics and then the IHC analysis was done. So you see that there is a positivity of the IDH protein, this mutated IDH protein. The positivity is usually in a nuclear and a cytoplasmic pattern. So the expression is present both in the nucleus and the cytoplasm. So the presence of IDH1 R132H IHC positivity shows that there is a mutation within the IDH gene, and you follow it up with analysis of the ATRX IHC, you see that the tumor cells are negative for ATRX. However, the internal controls, that is your endothelial cells, are showing expression of ATRX. So this particular tumor has got a IDH mutated, that is R132 mutation positive, and ATRX loss. And this fits in with the astrocytoma of the IDH mutated category. So how do the oligodendroglioma cases differ from the IDH mutated astrocytoma? While The IDH mutant astrocytomas show loss of ATRX. The first one is a grade 2 tumor. The second one is a grade 3 tumor. ATRX is lost in both the cases, while the oligodendroglioma cases will show retained ATRX, as you can see over here. So this is a oligodendroglioma in the third case. Let's look at this particular case now. This one, at first glance, it looks pretty much like a oligodendroglioma, right? You have got this characteristic round monomorphic looking nucleus with the perinuclear cytoplasmic clearing with the fried egg kind of an appearance. And you do IDH R132H IHC, and you see that there is a positivity of IDH. However, ATRX shows there is a loss of expression of ATRX with your internal controls in place. That is, your endothelial cells are showing the positivity. So even if morphologically this thing fits in with the oligodendroglioma, your final diagnosis will be a IDH mutated astrocytoma, because you have got a positivity for this mutated IDH protein and a loss of expression of ATRX. And usually in these cases of IDH mutated astrocytomas, you also get an upregulation, increased expression of P53. So that kind of goes hand in hand with that. This, however, is a case which is a proper oligodendroglioma. So these are your oligodendroglioma cells, which are obviously showing the atypia, and on addition to the atypia within the oligodendroglial cell, within the oligodendroglial cells, you also have in figure B microvascular proliferation, thus upgrading your diagnosis to a oligodendroglioma of grade 3. In the olden days, this would have been called anaplastic oligodendroglioma. We know that we don't use the terminology anaplastic anymore, so we'll call it oligodendroglioma, IDH mutated, and 1p19q co-deleted. But obviously, you know that in order to give this standard nomenclature, you will have to do a FISH or a NGS or a promoter methylation assay in order to reach the diagnosis, in order to reach the particular diagnosis. So how do these cases differ from each other as far as the IDH and the ATRX expression pattern is concerned? The first two cases, the first and the last cases are basically oligodendroglial, whereas the second case is a astrocytic tumor. We see that the IDH is mutated in all the cases, and the mutant IDH protein is expressed in all the cases. However, ATRX expression is retained only in the case of oligodendrogliomas, while it is lost in the case of the IDH mutated astrocytomas. So that's how you will differentiate these two important categories of tumor. Your astrocytoma IDH mutant will show positivity for IDH R132H protein, while it will not show expression of ATRX. Your oligodendroglioma will show expression of IDH1 protein mutant and will show retained expression of ATRX. We have also got a third subtype of tumor, that is the glioblastoma of the IDH-wildtype, which does not show IDH mutations because it is wild type, and will show retained expression of ATRX. And lastly, in order to give the diagnostic nomenclature of oligodendroglioma, you will obviously have to assess for 1p19q co-deletion, and this can be done by either FISH or next generation sequencing. And this is how FISH helps. So basically, you see that there is a loss of the red signals in both the 1p and the 19q locus. So there is a 1p19q co-deletion, and the deletion of 1p results in loss of tumor suppressor genes like FUBP1, and deletion of 19q results in loss of tumor suppressor genes like CIC, resulting in generation of the oligodendroglioma. Then the third abnormality is the TERT promoter mutation, also known as the telomerase reverse transcriptase. So this kind of activating mutations will result in increased telomere lengthening. As a result, the cell gets immortalized. And at the same time, because of the mutation, there will be increased binding of the transcription factor NF-kappa beta through the P50 and the P65 subunits. There will also be increased binding of other transcription factors like TCF, which is present in the WNT pathway, resulting in upregulation of the NF-kappa B and the WNT pathway, and cell proliferation and stem cell renewals, resulting in gliomogenesis. So you see over here, you have got these mutations at two sites. One around 124, 124 base pair apart. Before the promoter site, that is the site of transcription of the gene, and 146 base pair above. This altered site, this mutated site, has an increased tendency to bind the transcription factors, in this case the TCF transcription factor, resulting in increased upregulation of the TRT, telomerase, reverse transcriptase, resulting in all the kinds of gliomogenic events that we have seen. And these mutations can be picked up when you do when you do a mutational analysis, and you will pick them up in the form of the double picks when you see your chromatograms. The last genetic abnormalities that we shall be talking of are the CDKN2A and CDKN2B homozygous deletions. So basically these two genes are tumor suppressor genes, and the CDKN2A locus and CDKN2B locus are associated with the production of tumor suppressors. P16 and P14 are the two tumor suppressors which are encoded by the CDKN2A locus. P14, by stabilizing the P53, results in the block at the G1S stage of the cell cycle. On the other hand, the P16, which is encoded by CDKN2A, and P15, which is encoded by CDKN2B, inhibit the cyclin D CDK4 combination, as a result of which the RB pathway gets blocked and the cell gets arrested in the cell cycle. Now, obviously, if there is a homozygous deletion of these two particular loci, you will have loss of these tumor suppressors, and hence the presence of CDKN2A and CDKN2B homozygous deletion, that is both your copies are gone, has got a very poor prognostic significance, which is why we say that when in this molecular age, if you have a glioma which looks low grade, and if you have CDKN2A and 2B homozygous deletion, you have obviously you are dealing with a grade 4 neoplasm most likely. So this homozygous deletion of the CDKN2A and 2B can be picked up with FISH. As you see over here, this is your normal cell where you have a red and the green signal, and where they are fused, they are giving you the yellowish appearance, while all the other cells are having an absence of the red signal. So over here you have only the green signals which are associated with the chromosome enumeration probe, while those signals, the red signals which will help you to identify the presence of the CDKN2A locus, are not there. So this is the utilization of FISH in the assessment of the deletion of CDKN2A and 2B. However, the same thing could also be addressed by your DNA promoter methylation assay in combination with next generation sequencing. Over here, you see this particular case is a glioblastoma of the IDH-wildtype, which is associated with multiple amplifications and deletions. So you have got amplifications of the MD, M4, and the EGFR, that is the oncogenes. Those particular loci are upregulated and amplified, while there is a loss of there is a homozygous deletion of CDKN2A and 2B locus, which is picked up in the methylation profiling. So the importance of the presence of this CDKN2A and 2B deletion is that you might have a tumor, you might have a IDH mutated glioma which looks morphologically like WHO grade 2 or grade 3. But if you were to do NGS or FISH, and if you were to come across this particular finding, your diagnosis would change to a WHO grade 4, irrespective of whether you have the standard histological features of necrosis or microvascular proliferation. That's one way in which molecular genetics has made big inroads in the field of diagnostic neuropathology, because now the diagnosis is not completely histological. Molecular genetics has got a big role to play. So this is the snapshot of the IDH mutated astrocytomas. If you have just astrocytic atypia, it will be of grade 2. If you have increased mitotic activity, it becomes grade 3. And if you have the grade 3 features in combination with findings like microvascular proliferation and or necrosis, it becomes grade 4. But an important thing is that in a grade 2 or a grade 3 IDH mutant astrocytoma, if you have coexistent homozygous CDKN2A and 2B deletion, you will have to upgrade your grade 2 or grade 3 tumor to a CNS WHO grade 4 tumor. And these cases might actually sometimes morphologically mimic oligodendroglioma. So keep that in mind. And how will IHC help you? Obviously, in those cases, because it is IDH mutant, most of the cases will show a positivity for the mutant IDH1 R132H protein, and there will be a loss of expression of ATRX. However, remember that around 10% of the cases might show an absence of IDH1 R132H IHC, in which case, if the age of the patient, and in that case, you will have to basically assess the IDH gene, that is IDH1 and IDH2 genes, by DNA profiling. So that used to be done by Sanger sequencing priorly, but I think slowly over time, this particular niche will be taken over by diagnostic methodologies like next generation sequencing. Another important thing is that in such IDH1 R132H mutant, in those cases, you will also have to exclude the presence of the histone gene mutations, because some of those cases which lack expression of IDH1 R132H might actually be histone gene mutation associated tumors. So now this second case, obviously this is an oligodendroglioma that we are dealing with. I won't go over the morphology. You know how it looks. And in the olden days, that is back in the times when I was doing PG, it would have been called an oligodendroglioma, WHO grade 2 in Roman. However, at present, however oligodendroglial it might look, you will have to address the issue of IDH mutation, ATRX presence or absence, and 1p19q co-deletion in order to reach this final diagnosis. And if you do all those things, you will basically give out a diagnosis of oligodendroglioma, IDH mutant, 1p19q codel, grade 2. Sorry. So this is the hyper view of the same. This case of oligodendroglioma, however, shows some findings which are much more sinister. So towards the right-hand corner, you have an area which is hypercellular, which looks more blue, being composed of more diffuse mass of cells. And obviously, because there are more number of cells, you will most likely get increased mitotic activity in these areas. Along with that, you get this, this clustered congeries of vessels, which look slightly larger than a vessel and slightly more cellular than a normal vessel. So in the olden days, this would have been called anaplastic oligodendroglioma, WHO grade 3. Again, we shall have, in this present era, we shall have to assess for ATRX, IDH1, and the 1p19q co-deletion assay, and give a diagnosis of oligodendroglioma, IDH mutant, 1p19q codel, grade 3. Observe that the anaplastic terminology has gone out of the window, so we will not use that term anymore. So these are the grade 2 areas in this particular tumor, while these are the grade 3 areas. Obviously, you see there is a higher cellularity and there is increased mitotic activity that you are spotting over here. And also a bit of microvascular proliferation. You see one lumen, you see another lumen, and in between you have got a lot of proliferation of vascular endothelial cells. So you have got a lot of microvascular proliferation. So this is basically grade 3 oligodendroglioma. So the snapshot of oligodendroglioma, you will have this classic uniform round cells with fried egg appearance and your chicken wire capillaries. So this is the grade 2. In the case of grade 3, there is no grade 4 in the case of oligodendroglial tumors. You can use a constellation of findings to establish your diagnosis of CNS WHO grade 3 oligodendroglioma. So they will have increased cellularity, atypia, mitotic activity of at least 6/10 hyperfields, microvascular proliferation, and or necrosis. You might even have pseudo-palisading necrosis. And sometimes they will mimic astrocytoma of the IDH mutant and WHO grade 4 categories. How will IHC help? Keep in mind that GFAP, unlike your astrocytomas, oligodendrogliomas usually lack GFAP expression. However, there is a population of mini gemstocytes and gliofibrillary oligodendrocytes, which will show small bellies of GFAP positivity. Most importantly, the IDH1 R132H protein will be expressed in both the cytoplasm and the nucleus, but unlike the IDH mutated astrocytomas, nuclear ATRX will be retained in the case of oligodendrogliomas. And in those cases in which you are unable to get IDH1 R132H positivity, you will probably have to consider a gene sequencing analysis for your non-canonical mutations, or for your IDH2 mutations. And also you'll have to perform 1p and 19q co-deletion assay, which can be FISH, or slowly probably this will get changed over to NGS and DNA promoter methylation studies. What about CDKN2A and 2B deletion? So coexistent homozygous CDKN2A 2B deletion may help to upgrade the diagnosis of grade 2 to a grade 3. However, the relationship of CDKN2A and 2B homozygous deletion to upgrading something from grade 2 to a higher grade is not as robust as in the case of IDH mutated astrocytomas. Coming to the third category, that is the glioblastoma of the IDH wild type, which obviously has a grade of 4. Morphologically, this could be any kind of combination. So you can have a high grade astrocytic looking tumor with areas of necrosis, which could be of pseudopalisading or geographic nature of microvascular proliferation. But most importantly, sometimes you need not have those findings. You need not have necrosis. You need not have microvascular proliferation. You could just have an atypical astrocytic tumor, which could be of grade 2 and grade 3 histomorphologically. But if you were to submit it to next generation sequencing and DNA promoter methylation assays, if you came up with things like a TERT promoter mutation, or maybe an amplification of the EGFR gene, or a gain in chromosome 7 or a loss in chromosome 10, your histologically established diagnosis of a grade 2 or grade 3 would get upgraded to a grade 4 IDH wild type glioblastoma. And most important thing is that these cases are IDH and H3 wild type. That is, they will not show you IDH mutations, and neither will they have histone gene mutations. So this is how your copy number variation profile will help, basically. So the first case, both the cases are actually IDH wild type glioblastomas. However, the first one gives you the impression of a grade 2 tumor. The one which is after this has basically got a histomorphologically grade 4 kind of an appearance. But the important thing is this: the cases looked kind of different histologically, but once you did the DNA promoter methylation assay, you showed that there was a gain in chromosome 7 and a loss in chromosome 10 in both the cases. And as a result, your diagnosis gets changed over to a IDH wild type grade 4 glioblastoma in both the cases. And that's how DNA promoter methylation study, or NGS, is important in the diagnostic workup of these particular tumors. So as you see, these are the copy number variation plots which are generated by the DNA methylation profiling array data. And so this is the gain of the chromosome 7, and this is the loss of the chromosome 10. Observe that some of these cases will also be associated with a homozygous deletion of CDKN2A and 2B loci, but in the context of typical glioblastoma of the IDH wild type family, this does not have a prognostic significance, unlike in the IDH mutated family of astrocytic tumors. And there can be some IDH wild type glioblastomas which will be MYCN amplified. So this is picked up by your DNA methylation profiling as an increased amplification of the MYCN locus. So these are the various genes which are associated with the amplification or the deletion. So basically, you have got a lot of oncogenes like PDGFR, EGFR, CDK6, MET, etc. You also have the tumor suppressor genes in the form of RB1, P53, which once they're down regulated, will result in gliomas. And all these copy number variations of these genes can be picked up with your DNA promoter methylation assays. So in this particular case, this was a diffuse, there is a grade 2 astrocytoma, which was of IDH wild type. But once you did a DNA promoter methylation assay, you could show that there is a gain of chromosome 7 and there is a loss of chromosome 10, thus changing your diagnosis onto a molecular grade 4. So your final diagnosis from your grade 2 actually becomes a grade 4, that is a glioblastoma IDH wild type grade 4. So that's all about this particular family of tumors. They are wild type for both IDH as well as the histone genes, and naturally they are CNS WHO grade 4. They have to have a combination which can include your histological findings of microvascular proliferation necrosis. But even without that, if you had third promoter mutations or EGFR amplifications or gain in chromosome 7 or loss in chromosome 10, you could reach a diagnosis of glioblastoma, IDH wild type, grade 4. And you can have various morphologies over here. You will have the conventional morphology, but you could also have the small cell-like appearance. You could have a primitive neuronal kind of a morphology. You could have the granular cell appearance that we saw priorly, and you could have some deceptively circumscribed forms within this particular family. Most important being the giant cell glioblastoma and gliosarcoma, which can present in a superficial location and be very well circumscribed and can mimic either a METS or a meningioma. And the epithelial glioblastoma also belongs to this particular family and is also sometimes circumscribed and often shows mutations in the BRAF gene. So it's important. Which patients should you analyze by gene sequencing if you come across a glioblastoma of the IDH wild type? The rule is that if the patient is more than 55 years age and has got a history of less than 6 months duration, and if you are showing that IDH R132H IHC is negative and ATRX is retained, then most likely this is not a IDH mutated category. So in that case, you probably do not need to go in for a genetic sequencing. However, in those patients who are less than 55 years of age, who have got a longer duration of history, that shows that probably this particular tumor has evolved from a lower grade glioma. And in those patients in which the nuclear ATRX expression is lost, definitely there is a suspicion that there might be a IDH mutation, and in that case you will have to perform a gene sequencing for non-canonical mutations in IDH1 gene as well as the IDH2 codon 172 locus. Another important thing to keep in mind is that you also have to do a genetic sequencing to exclude the histone gene mutations in this particular family of tumors. Now coming to this particular tumor, this gives you the impression of a very primitive small round cell tumor, right? It's just sheets of cells which obviously have got a high mitotic activity and forming a diffuse mass, and overall giving a small blue cell tumor look. And in a young patient, you would probably suspect something like a small round cell tumor which belongs to the medulloblastoma group maybe. And then you have got these areas which look a little bit like Homer-Wright rosettes. You have got the molding of those primitive small round cells, and you have got these areas where there looks like there's a bit of Homer-Wright rosette like differentiation, and there's a bit of neurofibrillary matrix within this area. But in this present day, before reaching that particular conclusion, you will have to have lots of different things. I mean, lots of different entities in your mind. Like, for example, if it is in the midline, you will need to ask whether this case could be a diffuse midline glioma of K27 altered, which gets a grade of 4. If it is in the cerebral hemisphere, you will need to ask yourself whether it is a diffuse hemispheric glioma, H3G34 mutant, which also gets a grade of 4. It could be a astrocytoma of IDH mutant category, again grade 4. And if those mutations are not present in a young child or even in a teenager, this might be a diffuse pediatric type high-grade glioma, which is wild type for both the histone mutations as well as the IDH mutations, and that also gets a grade of 4. Of course, along with those various astrocytic gliomas, you will have your other differentials like medulloblastoma, a small cell variant of an atypical teratoid rhabdoid tumor, or even maybe a high-grade non-Hodgkin lymphoma, like a lymphoblastic lymphoma or something. So you have got multiple types of tumors which can present with the same kind of morphology. And this is where the role of IHC and the genetic sequencing is really important, because it helps you to segregate between these different tumor categories. So now we talk about the histone gene mutation associated gliomas, and the histone gene, basically, that is the histone proteins have got 3 variants. So you have got the 3.1, 3.2, and 3.3. So we shall take away those 0.1, 0.2, 0.3 things, and mostly deal with this H3. So basically, we are talking about the histone 3 gene mutations, and these mutations could be of K27M or G34RV. We shall be talking about those specific mutations and what happens in these mutations in the coming slides. But what is important is that in the pediatric age group, you can have IDH mutations, but the histone gene mutations are a very important cause of pediatric gliomas, and these need to be kept in mind, especially if you are dealing with a glioma in a young adult. So what are the role of the histone proteins? Histones are basically a family of proteins around which the DNA is tightly coiled, and this helps to organize the structure of the chromosome. And the histone protein molecules could actually be linked to different kinds of moieties. So for example, it could be linked to an acetyl group, or it could be linked to a methyl group. So acetylation of the histone would basically turn on the gene and resulting in increased expression of the gene, while the methylation would help to suppress the gene. So in case of DNA methylation, what happens is that these methyl groups are added to the promoter sequence, that is the CpG islands in the promoter sequences, and this in turn will result in recruitment of multiple co-repressor complexes like histone deacetylase and histone methyltransferases, and this in turn will result in the silencing of the transcriptional machinery. So let's talk about the various kinds of gene mutations and the role of the normal histone proteins in that particular context. Let's start with the left. So you have got the histone 3.3 variant, which is encoded by the gene H3F3A. And in this particular protein, you have got the at K27 position, you have got the amino acid lysine. It is acted upon by histone acetyl transferase. And as a result, this particular histone becomes acetylated histone, that is H3K27AC. And this acetylated histone is basically a transcriptional enhancer, and this will result in increased transcription and activation of multiple oncogenes like PDGFRA, cyclin-dependent kinase 4, 6, MYC, etc. On the other hand, the same histone protein can be acted upon by another enzyme known as histone methyltransferase, which is basically a complex of PRC2 protein with EZH1 and 2. And the lysine, which was priorly we have talked about at the position K27, is acted upon by this enzyme and is converted into the methylated histone, starting from monomethyl to dimethyl to trimethyl. This trimethylated histone, that is H3K27Me3, is very important because we have got an IHC marker for this particular protein. Now, what happens in the case of the H3K27 mutated gliomas? In the case of H3K27 mutated gliomas, the abnormal protein which is generated because of the mutation is called H3K27M. And again, you have got an IHC for this particular protein. What it will do is that it will bind to the PRC2 protein and not allow it to methylate the histone molecule. As a result, with increased expression of the H3K27M, there is a subsequent reduction in the H3K27Me3. That is, the trimethylated histone expression will go down the more there is an expression of the H3K27M mutant histone molecule. The same thing could also be mediated by another protein known as IZIP, and this IZIP also inhibits the histone methyltransferase, resulting in reduced production of trimethylated histone. So there will be a reduced expression of the trimethylated histone in both the H3K27M mutation and IZIP overexpression. Both these cases will be associated with a particular tumor phenotype known as diffuse midline gliomas. So a little bit about the diffuse midline glioma. As I said, the diffuse midline glioma is associated with a certain mutation in the histone 3 at the K27 position, wherein the altered histone, that is the mutated H3K27 histone, results in a reduced expression of H3K27Me3. And both the expression of this abnormal histone and reduction of the normal trimethylated histone can be detected by IHC. And as the name suggests, this is a midline glioma, so it will involve the midline regions of pons, thalamus, cerebellum, and the spinal cord. especially in the pediatric age group, but this age group will stretch on to the adolescents. So keep that in mind. And this could look like anything. This could look like your grade 2 to grade 3 astrocytic tumors. They could also mimic the highly cellular, even small round cell tumor kind of appearance, and could mimic WHO grade 4 glioblastomas. So the morphology does not really matter. What's more important is the status of the mutation in the histone genes. And irrespective of whether these have got a low grade or a high grade histomorphology, all of these cases have got a poor prognosis, and hence this family is given the designation of CNS WHO grade 4. So that's what happens with this particular mutation. At the K27 position, the lysine amino acid is replaced by methionine. So this is what is seen in the K27M mutation. As a result of this, the mutant protein, there will be increased expression of H3K27M mutant protein, and there will be a global reduction of H3K27M trimethylated histone. And because of this, there will be a consequent gain in the acetylation. And because of the acetylation, there will be increased oncogenesis because of increased activation of the oncogenes, resulting in the final tumorogenetic pathway. Now let's look at this particular case. This is obviously a high-grade glioma, which is present within the cerebellar white matter and the cerebellar peduncle. So, and obviously you've got a highly pleomorphic mitotically active astrocytic tumor, and then you have got a microvascular proliferation. So this has a high-grade morphology, but more important thing over here is to analyze for the H3K27M, and the altered mutant protein is expressed by these tumor cells, and hence the diagnosis becomes H3K27 altered diffuse midline glioma, CNS WHO grade 4. In this particular case, you have got a slightly enhancing tumor situated in the region of the pineal gland. This one has got a low-grade morphology. As you can see, it looks like grade 2 to 3 kind of an appearance. Grade 2 kind of morphology. And before giving it a diagnosis of a grade 2 astrocytoma, once you do a histone IHC for H3K27M, you see that there's a nuclear expression of H3K27M, and hence your diagnosis becomes diffuse midline glioma, H3K27 altered, CNS WHO grade 4. In this particular case, there is again a midline glioma, which has happened in the region of the third ventricle. Obviously, histologically, you will think of an oligodendroglioma, and there, because of some amount of microvascular proliferation, you will be tempted to call it a grade 3 oligodendroglioma, but obviously you will have to do an entire workup of your IDH, ATRX, and 1-peak 19q core deletion. However, all these cases will turn out to be negative in this, but all those tests will turn out to be negative in this case. And since you have done H3K27M IHC, you see that there is an expression of this mutant protein within the nucleus of the tumor cells, and hence your diagnosis becomes diffuse midline glioma, H3K27 altered, CNS WHO grade 4. This is an interesting case where obviously the first tumor, as in A, gives you a very low grade kind of a pyloid appearance with these Rosenthal fibers, and as a result, this was initially diagnosed as a pilocytic astrocytoma. The case later recurred as a cellular higher grade glial neoplasm, which had increased mitotic activity as well as is shown by Ki67 index. And once you did H3K27M IHC, you saw that this mutant protein was expressed in the tumor cell nuclei, and hence your diagnosis has to be revised to a diffuse midline glioma K27M mutant. So the prior diagnosis was wrong, and this is one of those diffuse midline gliomas which can morphologically mimic even pilocytic astrocytomas. This is obviously a higher grade astrocytic tumor which shows H3K27M mutant histone positivity, so it goes into the diagnostic category of diffuse midline glioma, H3K27M mutated. So you could have variable kinds of morphology. Like for example, here you have got an epithelioid morphology. Over here, you have got an appearance which looks like a pinnet. We don't use the term pinnet anymore in the context. But anyway, in both the cases, you did ISC for the mutant histone H3K27M, and you showed a positivity. And hence the diagnosis in all these cases is diffuse midline glioma. associated with K27M mutation. Now, the importance of assessing for this particular mutation is that it helps to segregate the morphologic mimics. But before that, let's talk a little bit about the other histone that we talked about initially. That is the trimethylated histone. I've already told you that with increased expression of the H3K27M mutant histone, you have a corresponding reduction of the normal trimethylated histone. So let's look at picture D and C. In picture D, you see there is an increased expression of the mutant histone H3K27M, and as a result, we know that with the expression of the mutant histone, there will be a corresponding reduction in the normal trimethylated histone. So the same case shows a complete absence of K27 of H3K27 trimethylated histone. So trimethylated histone is gone, while the K27M mutant histone is upregulated. Your endothelial cells serve as control over here. In the other case, the case which is shown in A and B, this is basically a histone gene wild type, meaning that this particular case there is no mutation of the histone gene. So obviously H3 K27M IHC shows negative result because there is no histone gene mutation, and because there is no histone mutation, there is normal expression of the trimethylated histone which is expected in a normal cell. And so you see that a combination of H3K27M along with H3K27 trimethylated histone ME3 gives you the best result when it comes to analysis of this particular group of tumors. This is important when we talk about the morphologic mimics of this particular tumor. As I said, these tumors can look very, very highly cellular and like a small round cell tumor. As a result, differential diagnosis like medulloblastoma or atypical teratoid rhabdoid tumor of the small cell morphology could come into a differential diagnostic workup. But obviously, if you do a combination of H3 K27M mutant histone along with H3 K27 trimethylated histone, you will see that the results are exactly the opposite of what you see in the case of diffuse midline gliomas. So there will be no expression of this mutant histone, H3K27M, while there will be normal expression of trimethylated histone, as you see. So trimethylated histone is expressed in both the cases, while there is no expression of the H3K27M mutant histone in these morphologic mimics. So the histone H3K27M IHC along with H3K27 trimethylated histone IHC is very important in the workup of these tumors. Similarly, you could have an astrocytoma of the IDH mutated category or an oligodendroglioma of the IDH mutated category, which might mimic diffuse midline gliomas. In those cases also, you will see that there is a normal retained expression of H3K27 ME3, while there will be a loss of there will be no expression of H3K27M. So H3K27M is very important in the differential diagnostic. Similarly, in the case of this grade 3 astrocytoma of the IDH mutant category, which is shown in E and F, you have expression of the trimethylated histone. You don't have any expression of H3K27M because it's not present, this particular mutation. In a case of this glioblastoma IDH-wildtype, again, you have got retained expression of H3K27 trimethylated histone, no expression of H3K27M mutant histone. But there are two other important categories of diffuse midline gliomas, and these two categories are not associated with H3K27 mutations. So this one of them is called the EGFR mutant category. The other is the AZIP overexpressed category. And if you were to do a DNA methylation profiling, you could see that these categories can be segregated from each other. Now, coming to the diffuse midline gliomas, which do not show histone mutations, but are associated with AZIP overexpression. Like I already told you, overexpression of AZIP is associated with downregulation of the trimethylated histone production. Right. So basically over here, the first H&E image shows you a low grade, kind of a grade 2 astrocytic morphology. If you do H3K27 ME3, that is trimethylated histone, you will see that the tumor cells are all negative for this particular protein, meaning that there is a absence of methylation of the histone molecules. Now that usually happens because of H3K27M mutant protein, right? But in this case, if you do a H3K27M mutation IHC, you see that this particular protein is absent. Why? Because this role of the mutant H3K27M is actually taken over by AZIP in these cases. And the AZIP protein is upregulated within this particular tumor cells. And this AZIP in turn leads to a loss of expression of the trimethylated histone. So these cases of diffuse midline glioma, you will not get H3K27M mutation, rather you will get AZIP overexpression. And AZIP IHC can be utilized in order to segregate this particular family of tumors. So let's analyze these two cases. The first case is a diffuse midline glioma, that is ABC. This first column is associated with AZIP overexpression. So if you analyze for H3K27 trimethylated histone, you will see that your tumor cells are all negative. If you assess for the mutant H3K27 protein, you will see that the tumor cells are not showing any expression of H3K27 protein. However, they are showing a overexpression of AZIP. On the other hand, the lower column is basically what is seen in the case of the standard diffuse midline gliomas, which are associated with H3K27 mutation. These cases also will show no expression of H3K27 trimethylated histone. They will, however, show the presence of the H3K27 mutant protein, and AZIP will not be present in these cases. The other category of tumor within this particular family is diffuse midline gliomas of the EGFR mutant category. These cases are also associated with loss of expression of the trimethylated histone. But when you analyze these cases for IHC with H3K27M IHC, you see that there is no expression of H3K27M. So in these cases, actually, your tests like next generation sequencing or promoter methylation assays come in handy in order to diagnose this particular group within the diffuse midline glioma family. Coming to the diffuse hemispheric glioma family, the diffuse hemispheric glioma, as the name suggests, occurs within the cerebral hemisphere. And what is the genetic abnormality that is seen over here? So let's analyze this, this particular genetic abnormality. You have got G34, R on top, and V. Okay, so at the amino acid position 34, the normal amino acid is basically glycine. Because of the mutation, this could get changed over to either arginine, which is R, or valine, which is V. So this kind of mutations within the histone gene H3F3A will result in reduced production of trimethylated histone, which is called K36Me3. And as a result, this will result in upregulation of the proto-oncogenes. Now, the important thing is this G34RV issue can be utilized for IHC. How? This G34R and G34V are the mutant proteins, and both of these can be actually detected by IHC. So immunohistochemistry for those mutant proteins can be utilized to diagnose this group of high-grade cerebral hemispheric gliomas. And this usually happens in the case of adolescents to young adults. And most importantly, they can resemble your medulloblastomas. They can look like embryonal tumors. And to make things even worse, they can actually sometimes lack GFAP, and even OLIG2 can be negative. So those are basically glial markers, right? So sometimes those glial markers can be negative in this particular family of tumors. Another important thing to keep in mind is that these will often be associated with loss of ATRX expression. Like I said before, ATRX expression is mostly associated with IDH mutated gliomas, right? But in this particular family of tumors also, you could have a loss of ATRX expression and a diffuse P53 staining, which could be utilized to diagnostic advantage. Irrespective of how they look, these cases will all get the designation of CNS WHO grade 4, just like your diffuse midline gliomas. So what happens is that because of this particular mutation, there is inhibition of the SETD2 protein, as a result of which there is the block in conversion of K36Me2 to K36 trimethylated histone. And as I said, you have IHC. So in this particular case, this particular mutant protein G34R is seen to be expressed by these tumor cells, while your endothelial cells act as the negative internal control. The endothelial cells are negative for this particular protein, while the case below is actually a wild type, is a wild type tumor where there is no mutation of this kind, and as a result the tumor cells are all negative. So let's see this particular case where you had a small glioma within this cerebral hemisphere, and morphologically speaking it gives you the kind of a small round cell kind of morphology. And once you analyze the tumor in a sequence, you see that some important findings come out, right? First of all, the expression of GFAP is patchy, focal. There is a complete absence of OLIG2, which is basically a glial marker. So this OLIG2 is not expressed. P53 is upregulated in the tumor cell nuclei, and ATRX expression is lost. So that's an important thing that I said can be seen in this particular family of tumors. You analyze the case for the mutant H3G34V and see that the tumor cells are all expressing this particular mutant protein. And just for completion's sake, you do a H3K27M IHC as well. Obviously, this is not a diffuse midline glioma, so this is negative. H3K27M is negative. In this particular case, again, you have got a hemispheric tumor, which gives you a small round cell that is something akin to medulloblastoma morphology, the primitive embryonal appearance. OLIG2 is again negative in these tumor cells. So sometimes establishing the diagnosis of glioma becomes challenging in these cases because glial markers may not be expressed. P53, like I said, is diffusely upregulated within the tumor cells. And also, if you do a mutant IHC protein, if you do a mutant H3G34R IHC, you will see that the tumor cell nuclei are showing positivity for this mutant protein. And like I said, some of these cases, quite a few of these cases will show a loss of ATRX expression, which is also seen in this particular case. So in the context of the pediatric high-grade gliomas, we have talked about the IDH mutated cases. We have talked about the diffuse midline glioma where there is a K27 mutation. We have talked about the hemispheric gliomas with the G34 mutation. There is a category of high-grade pediatric high-grade gliomas, which are WT stands for wild type, which are wild type for both histone gene mutations as well as IDH gene mutations, and these are basically associated with copy number variations of EGFR, PDGFRA, MYCN, CDK6, etc. So this is the snapshot of these high-grade pediatric gliomas. Most of them will actually resemble glioblastomas, but some could actually mimic the embryonal CNS neoplasms like medulloblastomas. All are of the WHO grade 4 category, except for a family of tumors which has been newly identified by DNA methylation profiling, and this family is known as the infant type hemispheric glioma. It has not been given any WHO designation of a grade, and these are all IDH wild type cases. That is, they do not show IDH mutations, they show histone mutations or mutations in other genes. So we have talked about the diffuse hemispheric glioma and the diffuse midline glioma. So coming to the diffuse midline glioma, I said that the mutation is in the H3K27M in many of the cases, but some cases might not have the H3K27M mutation, in which case you will have AZIP overexpression. Some cases will not have that, and rather they will have the EGFR gene amplification. And OLIG2 is an important marker in this context because this will identify these glial tumor cells. Nuclear expression, in those cases in which the mutant H3K27M protein is present, there will be nuclear expression of it. Whereas in the H3 wild type cases, you will get AZIP expression. Correspondingly, there will be a loss of expression of the trimethylated histone. And this can sometimes lack ATRX, although most often they will have retained ATRX expression. In the case of diffuse hemispheric gliomas, this can be negative for both the glial markers of OLIG2 and the GFAP. This will be positive for the mutant proteins H3G34R and V. This will be negative for ATRX, and they will show diffuse P53 positivity. And obviously, the genetic abnormality is because of a missense mutation in the H3F3A gene, resulting in generation of mutant protein G34R or G34V. The other two important categories of high-grade gliomas in this family, in this particular group, are the diffuse pediatric high-grade glioma of the wild type H3 and IDH-wildtype. Like that is, this particular tumors lack mutations in both the histone genes as well as the IDH genes. So over here, the mutations actually are present in multiple other pathways like EGFR, MYC, and P53 pathway, etc. And here also the OLIG2 and GFAP expression might be focal, and neuronal markers can be expressed in the case of the MYCN subtype. And nuclear ATRX will be lost even in these particular cases. And the last category of high-grade glioma in this category is, in this group is, like I said, infant type hemispheric glioma, which can sometimes show positivity for ALK and EMA. So they might be utilized in the immunohistochemical context. There are multiple receptor tyrosine kinase gene fusions within this particular group of tumors, resulting in aberrant MAP kinase signaling pathway. So what is this MAP kinase signaling pathway? This is basically what we read in the Robbins oncology, right? So basically you have got a growth factor which binds to the growth factor receptor, which is coupled to the receptor tyrosine kinases, which on activation results in phosphorylation and activation of downstream molecules of RAS, RAF, MEK, and ERK. ERK is also known as MAP kinase, and hence this particular pathway is known as the MAP kinase pathway. This particular MAP kinase pathway will become important because in the next category of tumors that we are dealing with, that is the low-grade gliomas, some of them could be associated with MAP kinase gene abnormalities. So talking of the low-grade gliomas, let's start with the one which is the most interesting and which has come up as a new entity in the WHO 5th edition. At first glance, this particular tumor looks like a oligodendroglioma, right? You have got the characteristic round cells with the fried egg appearance. And anyone would probably first talk of an oligodendroglioma, and you have also got some prominent vascularity over here. But this is not an oligodendroglioma. This is actually a newly grouped family of tumors known as the polymorphous low-grade neuroepithelial tumor of the young, or in short, PLNTY, PLNTY. Now, this could look like oligodendrogliomas, but as the figures C and D show, they could sometimes look like your conventional fibrillary astrocytomas, right? So they could look oligodendroglial. They could also look astrocytic, this family of tumors. Is there an IHC help? Yes. So you could do a BRAF V600E IHC with the VE1 antibody, and you can show a cytoplasmic positivity, which would help to establish this particular diagnosis. Of course, that would not be a foolproof establishment. For that, you will need to do a DNA promoter methylation assay, but still IHC is of help. And CD34 shows a membranous positivity, which again helps in segregating this particular tumor from its morphological mimics like oligodendroglioma. And this particular family is associated with FGFR, that is fibroblast growth factor receptor gene fusion with multiple other genes of like TSCC, KIA1598, or CTNNA3. And if you do a DNA methylation profiling, you will see that this particular group of tumors segregates into its own category and clusters next to ganglioglioma, which, however, is morphologically different from that of PLNTY and can be easily segregated histologically from this particular tumor. The other family of low-grade glioma is something that we call an angiocentric glioma. Observe in the histology over here that you have got a very low-grade kind of a morphology. You have got a prominent vascularity with some amount of clustering of the cells just next to the vessels. You have got some entrapped neurons over here. And the overall appearance is a very low cellularity tumor of a low-grade morphology with an angiocentric kind of a profile. And at places, the cells look like they're arranged in perpendicular orientations next to the vessels. And this kind of gliomas with this characteristic angiocentric pattern, which can sometimes look like ependymomas, are the angiocentric gliomas, and these are characteristically associated with this particular MYB QKI gene fusion. So like in this particular case, you see that it's a low-grade fibrillary kind of astrocytic morphology with a perivascular pattern, as has been highlighted in this case. The tumor cells are not really very high grade. You don't see much of nuclear chromatin coarsening or nuclear contour irregularity. There is no mitotic activity to speak of in this particular cases. It's only the angiocentricity which is striking. And this angiocentricity could take the form of these elongated perpendicularly arranged cells next to the vessels. As a result, they could be sometimes mistaken for ependymomas. So you have got these ependymoma like perivascular processes. And at the same time, you also have, like in the third picture, you have this pyometer, and then just below that you have a clustering of these tumor cells in a perpendicular fashion. So you have got subpial clustering, which can be seen in these cases sometimes. And this is a grade 1 tumor, which is characteristically associated with MYB QKI gene fusion. And this particular fusion results in activation of the oncogenic MYB pathway and loss of the QKI tumor suppressor pathway, resulting in angiocentric glioma generation. So these are the various kinds of pediatric diffuse low-grade gliomas. As I've already said, the angiocentric glioma is associated with MYB alterations, and the polymorphous low-grade neuroepithelial tumor of the young or plenty is associated with MAP kinase pathway gene alterations. So how do you approach this case of pediatric gliomas? As you see, there are multiple different categories. Each has got its own unique molecular signature and sometimes immunohistochemical profile. So with the low-grade gliomas, at start, you could probably opt for a next-generation sequencing if you have an idea of the kind of tumors that you're probably dealing with. More often, you will find yourself testing for BRAF alterations, and especially for fusions of BRAF KIA gene. And BRAF V600E testing with IHC or PCR. You will have a H3K27M mutation testing, because some of your diffuse midline gliomas could actually look like a low-grade glioma. So you will do an IHC for H3K27M, and if you have it, also H3K27 trimethylated histone. And in some cases, you will need to opt for a gene sequencing of H3.3, 3.2, or 3.1 variants. And you will also need to do a IDH1 R132H mutation in order to exclude the slightly uncommon but still sometimes seen category of IDH mutated astrocytic tumors. In the case of the high-grade gliomas, diffuse midline glioma and diffuse hemispheric glioma are two important categories. So you will need to assess for H3K27M mutation testing, again by IHC and sometimes by gene sequencing, which can be done by the Sanger sequencing, but of late is being done by NGS at some places. For if it is in the cerebral hemisphere, you will need to assess for H3.3 G34R and G34V mutations. That can be done by IHC, or you could straight away go ahead by Sanger sequencing to do a H3.3 gene analysis, or consider next generation sequencing. If there is a recurrence or progression of the disease, then obviously the way to go would be to assess for multiple genetic abnormalities at the same time, and hence opt for a next generation sequencing. Consider a DNA methylation profiling in those cases which have got ambiguous features, so that you can get a comparison with the family of the known tumor cohorts, and which will give you a comprehensive diagnosis which is matched against standard reference samples. This is the last discussion for this particular chapter. So this is the tumor that I said initially. In back in the day, they would be diagnosed as oligoastrocytoma, right? Because you have got tumors which look slightly atypical astrocytic, but at the same time you have got those cells which look oligodendroglial at the same time. So, and if you do not have access to, like advanced methodologies like IHC or molecular genetics, you will probably give a diagnosis of this kind. However, keep in mind that this particular diagnosis has gone out of the window. We don't give this diagnosis, and you will have to analyze these cases for IDH, ATRX, and 1p19q code deletion at the minimum, in order to give a certain diagnosis which is aligned to the current WHO lineages. So here you have got some cells which look a bit oligodendroglial. You have got some cells which are much more pleomorphic to be called oligodendroglial, and they look astrocytic, atypical astrocytic cells. There is one cell over here which kind of looks like a mini gemstocyte, which again creates confusion. So these cells which I am circling right now are obviously too atypical to call an atypical oligodendrocytes. This fit in with the atypia within the astrocytic family. And Obviously, in the second picture, you have got a mitotic figure which is highlighted, so thus giving it a designation of grade 3. So what is the current diagnostic approach for this family of tumors? The first thing to do will be to check for the IDH mutation status. And how will you check for this mutation status? Your IDH R132H IHC. If it is mutated, well and good, go ahead and see for the expression of ATRX, which is the next thing to do. So if the IDH is mutated and ATRX is retained, as I've said, this fits in with the oligodendroglial kind of profile. However, in order to reach the diagnosis of oligodendroglioma, you will have to check for 1p19q co-deletion, right? And this is done by FISH, but can also be done by DNA promoter methylation assays or next generation sequencing. And if your 1p19q co-deletion is seen, then obviously your diagnosis is oligodendroglioma, IDH mutant, and 1p19q co-deletion. And based upon the histological criteria, what you are seeing, you will have to give the grade of 2 or 3. If, however, your IDH is mutated, but the ATRX is lost, then you are dealing with a IDH mutated glioma, astrocytoma. IDH mutated astrocytoma. IDH mutated astrocytoma could also be seen in the scenario where you have a IDH mutation, but where you have a ATRX retention and you do not have a 1p19q co-deletion. In both these cases, you will have to immediately check for the homozygous deletion status of CDKN2A and 2B by FISH over here, but again, slowly being taken over by NGS, DNA promoter methylation assay, etc. So if you have a CDKN2A, 2B deleted status, irrespective of whatever grade you might have histologically, this gets a diagnosis of astrocytoma IDH mutant, WHO grade 4. If it is showing retention, that is, there is no homozygous deletion, then based upon your morphologic diagnosis, you will give a diagnosis of astrocytoma IDH mutant grade 2 or 3. For those cases which are IDH mutated, ATRX retained, but with 1p19q intact, that is, there is no co-deletion, this also goes in with the uncommon family of astrocytoma IDH mutated cases. In these cases also, you will have to consider checking for a homozygous deletion of CDKN2A, 2B locus. And if you have a deletion of CDKN2A, 2B, obviously you have got a diagnosis of astrocytoma IDH mutant grade 4. If the CDKN2A and 2B is retained, check whether the case is showing the presence of necrosis or microvascular proliferation. If it is showing the presence of necrosis or microvascular proliferation, then again you can give a diagnosis of astrocytoma IDH mutated grade 4. If there is no necrosis, no microvascular proliferation, it gets the diagnosis of astrocytoma IDH mutant, and based upon the histological findings of your mitotic activity, either a grade of 2 or a grade of 3. So this is the approach for the IDH mutated family of astrocytic / oligodendroglial gliomas. The next slide is about the IDH wild type cases. Now keep in mind that for calling something wild type, you are saying that this particular tumor does not have a mutation in IDH, right? So obviously these cases need to be thoroughly worked up for IDH mutation testing. And that is in those cases in which the IHC fails to give you a clue, you will have to consider the genetic sequencing within the IDH locus as well, IDH 1 and 2. So after doing the genetic analysis for IDH mutation testing, if you find that these cases are wild type, you will then next need to keep in mind the possibility of histone gene mutations, right? And for that, first, you will have to separate your tumors based on whether they are situated in the midline or whether they are situated in the hemispheric location. Let's talk about the midline family of tumors first. So DMG, right? Diffuse midline gliomas, and we know diffuse midline gliomas are associated mostly with the H3K27 mutation, K27M mutation, sometimes with AZIP overexpression or with EGFR amplification, right? So the first thing to do will be IHC for the mutant protein H3K27M, and if you have it, also H3K27Me3 trimethylated histone. If you see H3K27M expression with loss of normal nuclear trimethylated histone, you reach a diagnosis of diffuse midline glioma H3K27 altered. However, if this approach fails, You will obviously have to keep in mind the possibility of a AZIP overexpressed diffuse midline glioma, right? So you will do an IHC for AZIP protein. And if AZIP overexpression is seen, you will call that a diffuse midline glioma of H3, that is histone gene mutation wild type. That is, there is no histone mutation, but there is AZIP overexpression, and then it will get a grade of 4. These cases usually will show retained expression of ATRX, but sometimes they may lack expression of GFAP. Another way to approach this case is without doing IHC would be probably to go in for a direct gene sequencing of 3.3, 3.1, and 3.2. However, that would be probably a wasteful approach, and most often you will find yourself going down the IHC way. But you could also do a genetic sequencing in order to isolate this family of tumors. Talking about the hemispheric located tumors, check out whether your tumor is showing the presence of nuclear ATRX or not. ATRX may be present or may be lost. Irrespective of this, you will do a IHC for the mutant G34R and G34V proteins, right? Because this is in the hemispheric location, and you would like to exclude the diffuse hemispheric glioma of G34R and R or V mutation associated diffuse hemispheric glioma. So you could do an IHC for those mutant proteins, or again, you could straight away go in for a histone 3.3 gene sequencing, either by Sanger sequencing or by next generation sequencing. So if your mutant H3G34 protein, either G34R or G34V expression is present, or if your gene sequencing result is confirmatory, you will give a diagnosis of diffuse hemispheric glioma H3G34 mutant of grade 4. These cases will often show loss of ATRX expression and diffuse P53 positivity. Keep that in mind. However, if there is no expression of these mutant proteins and gene sequencing also shows you that there is no mutation of H3G34 type, in the case of the adults, check whether there is microvascular proliferation, necrosis, TERT promoter mutation, EGFR gene amplification, or gain of chromosome 7 and loss of chromosome 10, in which case your diagnosis becomes a glioblastoma of IDH wild type nature. And in the case of the pediatric age group, and then this particular thing is mostly settled by DNA promoter methylation assay. Check whether there is PDGFRA or EGFR or this EGFR alteration or amplification of the MYCN or a characteristic DNA methylation profile, and you will probably reach a diagnosis of diffuse pediatric high-grade glioma H3, that is histone wild type, and also IDH wild type, which gets a grading of 4. And one important fact, you don't call the pediatric high-grade gliomas as glioblastomas. That terminology has basically gone out as far as the pediatric age group is concerned. So this is in short about the various kind of diffuse gliomas that we get in clinical practice, and how the new WHO classification has segregated them into various molecular and histological categories. As we see, a lot has changed from 2007 through 2016 to 2021. The picture is still evolving, and probably by the next WHO classification, we shall see much less contribution from histology and a much bigger role for molecular genetics to play. And with this concluding note, I would like to end this particular video. But before that, a little bit of a little bit of plugging for this particular book, which has been basically written by me, and it talks about the brain tumor pathology in the context of the latest WHO 5th edition. It's kind of a condensed pictorial notes with quite a good number of illustrative images, and I would suggest that you go in for the Kindle edition, which comes at a price of 99 rupees, which is a base price. And you will find quite a lot of detailed information over there in the form of high-yield bulleted notes, which might be useful a day before the exam if you have to, go through a vast amount of information in a very short period of time. And with that, I reach the end of this discussion. Thank you.